Okay, I think we should kick off. Hi, everyone. My name is Kainwen, and I'm one of the directors at Shine Cancer Support. Thank you so much for joining us for our keynote session today. I've just realized that my paper with all of the the introduction information has fallen on the floor, but we're really delighted this afternoon to be joined by Professor Andrew Biankin. Andrew is a director of the Wolfson Wall Cancer Research Center. He established the Glasgow Precision Oncology Lab, which has produced the Glasgow Cancer Test, which is now used worldwide. He has authored over 200 articles in major journals, including seminal works on cancer genomics and precision medicine. So we are absolutely delighted that he's joined us this afternoon from his office in Glasgow at home. This is not a Zoom background, by the way. But we're really happy to have you with us. Andrew, thank you so much for joining us.
My pleasure. Thank you very much for the invitation. I'm really looking forward to the next hour of how we interact. First, I'd like to put in a plug for Scotland. I am brew, now the drink of US Congress, based on AAC. So get some if you can. Rare outside of Scotland in the UK. So I'm going to start something a little different today. I'm going to start sharing my screen, and I'm going to start with a video, just to set the scene of where we're going and where we're moving to. Presume everybody can see that. Is that good, Cayman? So what can we do? What have we learned over the last decade or more, or the last 50 years, that can guide us into how we develop new drug treatments for patients? How can we prevent cancer better? Can we detect How can we detect cancer better? I'm coming at this not just from an academic perspective, and these are some of my disclosures. I work also in industry, founded a number of companies, mostly focusing on the areas of precision medicine, precision oncology, drug development. And how do we take the knowledge we've gleaned from all the great biology that we've done in the past into something that's going to develop new treatments? So cancer is a disease of the genome. It's not to say that it is a disease of the genome we're born with, although sometimes they can predispose like BRCA1 and BRCA2, etc., for breast cancer. But something goes wrong with the genes in our organs. And there's various different types, which I won't go into detail, but you may hear about some of these. And it's really just breaks in the genome or changes that are called a mutation that lead to different types of cancers. And our understanding of the genomes of these cancers, just like we know about the Human Genome Project, we're so different as individuals, cancers are also very different. They have some similarities, but in the way in the past we categorized cancer, it was just like this. Oh, okay, I've got cancer of the breast, it's all the same, we'll treat it the same way. But now, as we understand more about what underpins and drives these cancers, even though these cancers look exactly the same under the microscope, they can be vastly different based on their genetic or molecular composition. And you can see this concept of precision medicine, of targeting using the right drug for the right patient, is being proven by tamoxifen here, that targets the estrogen receptor. It's present in about 60 to 70%. And if people say precision medicine doesn't work, the best drug ever for cancer, saved more cancer lives and years of quality of life, is tamoxifen, which is an example of a targeted therapy of precision medicine. So what makes it so difficult for us to do that? Now, look at the degree of diversity we're seeing in these different subtypes. Even in the sort of triple negative subtype that you may have heard of, there's a lot of diversity as well. So we struggle, but when we target the right drug to the right thing, like we see in chronic myeloid leukemia, or in drugs that target the EGFR receptor in lung cancer, we see dramatic responses. And those people that say, well, no, we're not seeing a big difference, if you extrapolate every one of those differences where we match the right drug to the right patient, you get a dramatic response. Even with immunotherapy, you have to match the right drug to the right patient. And this is what we do now. We'll start with the one that's most likely to work. It might be 25% of patients respond. The second most likely is 18%, and the third most likely is 12%. But the right treatment for your patient or for you is drug number 3. And if you've got something like pancreatic cancer, you're usually dead before you get to number 2 or number 3. So the principle of precision medicine, precision oncology, is to test something. And now that we know so much more about the biology and what underpins cancer, then we can match the right drug to the right patient. I'll give a personal example of 2 friends of mine in their 40s, not that long ago, both diagnosed almost at the same time with triple negative breast cancer. One wanted to explore all options. And these were not people that were just in the lay public. One was a professor of cancer medicine, one was a professor of cancer diagnostics. So they all knew what the disease was about. One chose to explore everything possible, did all these tests, etc. And the other said, well, even if I do a test, I'm not going to be able to get a drug to be able to treat myself. So what's the point? The one that said, what's the point, was dead within 18 months. And the one that explored all these different options, things that were off-piste, not part of routine healthcare, not on the shelf, so to speak, she lived for 6 years, even longer. So how is it that we can do that in an individual basis? And you saw Leslie's quote there. She just happened to chance upon the right drug that targeted her tumor directly. So how do we go from a system where we're very generic and very broad brushstroke and protocol driven to somewhere we have a bit of nuance? We can see evidence of this across all cancer types. So we have more and more treatments to choose from with pancreatic cancer, which is something very dear to my heart. But each of these drugs will only impact on maybe 1, 5, at most 10% of patients with some sort of significant effect. So it's almost a no-brainer question. We know that each cancer is different, even if it's from the same organ, even though it looks the same under the microscope. Yet we treat them the same, and we hope that, okay, we'll treat a bit more, so we'll get a slightly better response that we have. But does that help us address an individual patient's question? It's the law of averages. Now, how long is this going to work? Well, you've got a 30% chance of working. Well, it's not really satisfactory in 2021, is it? And what sort of cancer do I have? Well, specifically, what is it? And we've made great inroads in some cancer types like breast cancer, but others we lag far and far behind. Which treatment do I choose? Well, which one's going to work? Will it work? And of course, how long have I got? And how do we answer these questions for the individual and not the average? And not only can we do this for just those that can access these things through connections or high net worth, how can we democratize this as access for everybody in every health system across the planet, irrespective of the personal wealth, socioeconomic class, or demographic? So what do we need to do? If we're understanding biology, we're understanding these differences, we need to measure these differences. It doesn't matter what it is, we just need to measure those differences because that's going to inform us. We need to try and get more and more data, not just do a clinical trial where you only test what you want and what you need and then hope that it'll work out, because there might be anti-genes that prevent the resistance genes that prevent your cancer from working. And remember, in clinical trials, we're only looking at 2 or 3 or 4 or 5% of patients. You saw that most clinical trials, most patients don't get onto clinical trials. 90+% of patients in the world get treatment, and we don't learn anything from them. So how can we embed this in the health system? We talk about this, and we've got companies like Flatiron, etc., doing this in a semi-broad way. But we're impeded through all sorts of different things, not just logistics, but also privacy legislations, those who are familiar with GDPR, etc. So how do we find the right balance between this? And how can we share data around the world so we can learn from each and every patient? Because otherwise, those cancers that are less common, we're never going to get a treatment for, because there's just not enough of them in any individual jurisdiction to run a clinical trial. And of course, the economics just aren't there. So that we need to find a better system to do this. So what we aim to do was to enable this, at least for pancreatic cancer to start with, and then moving to other cancer types. So in the Glasgow Precision Oncology Laboratory, we built a test that doesn't cost £3,000 or £4,000 or £5,000 that you have to send off to Boston or somewhere to get done. A test where the health service can deliver and analyze themselves and keep the data at a price point that can be affordable, that's affordable, that can seamlessly integrate with health services. Then we built a platform for pancreatic cancer called Precision Panc. And then we also lead the International Cancer Genome Consortium, where we take data from Precision Panc and other studies all around the world. We're talking about hundreds of thousands of patients. Japan, Korea, India, China, all contributing so we can learn about cancer and share that information appropriately. And it's a challenge. So the first thing we did was trying to build an affordable and fit-for-purpose genomic test that works seamlessly and integrated efficiently with the health system. So a group of us got together, and we had to do this because if we were trying to build a platform like Precision Panc, we simply couldn't afford to send £3,000 per patient to do the test. It was just impossible, so we worked out a way to do it. And what did we do? We defined what are the real genes we need to know about. What's relevant and what's important? Do we need to test for every single gene at 100 times the cost, or can we just do the genes that are directly relevant to cancer based on our understanding of cancer biology, the literature, etc.? It had to work for all cancers, or the majority of cancers. It had to be affordable, this is critical, and it had to be usable in the real world. And that's what we built. We called it the Glasgow Cancer Test. It's now distributed globally by our partners, Agilent, and it's really on a way to not just treating patients like we see in the red circle, but even capturing more genes that embeds research in the NHS. So we start to learn from every patient. So we need to learn from every patient, as I mentioned. So we built a platform called Precision Panc. Now, this is important for cancers that are below the top 4 in incidence and prevalence. So once you go below lung, colon, breast, and prostate, then it becomes difficult to do meaningful clinical trials of those molecular subgroups or the genetic subgroups that I spoke about in any one hospital or in one, even in a small, a group of hospitals. There needs to be a platform, there needs to be a network where all patients can have the option of a clinical trial. So we changed the patient pathways in the NHS to really offer every pancreatic cancer patient an option in Precision Panc. We believe that if you have your treatment at 2 years still has less than a 10% survival, you don't really have a standard of care. So there's opportunities there to move beyond just our current thinking for those patients and those diseases where you're really making a minuscule difference and you're stuck with the drugs that are going to give you a minuscule difference. And you have to go on those drugs that aren't going to make much difference, and you're probably going to die before you get to a drug that might work. So we need to relax that somehow. So how Precision Panc works, and this is a slightly busy slide, but it's worth looking at. Everybody gets the option of what's called a master protocol. What this means, it's an opportunity for patients to say, well, come and join us, come and join the platform. We have all these studies available for you, and if you're part of Precision Panc, we'll aim to find the right trial and the right treatment for you. And you can join it at any step. Most importantly, for both the patient and the doctor, is to think about this ahead of time. Think about this at the time of diagnosis, or even prior to the time of diagnosis when you're suspecting it, because you can't get all of these things done in a quick enough time in order to match patients onto a clinical study. So already, when you're on your first treatment, you should be thinking about the plan, what's going to happen, rather than getting the result going, oh, gee, that's, the cancer's progressing. Oh, dear, what do we do now? What do we do now? And there's a panic. Hope for the best, but always plan for the worst. And this is what this platform is designed to do. Patients are the most important aspect, and their families are the most important aspect of this. Nothing will happen without you guys. Look at the movements that are currently available in various countries. Access to new medicines, pressure to access new medicines, balanced with the overall quality of life, all these aspects. And they won't listen to us. They need to listen to you guys, and they will listen to you guys. And here's a couple of examples of things that have happened, and still the system doesn't change. So this is Barbara. Barbara was told she can't access a drug in the NHS for a liver cancer or colon cancer that moved to the liver. She said, well, I can go buy this drug somewhere else. So when she went off and bought this drug, spent £21,000, and guess what? It shrunk the cancer in her liver. And it was then you could remove it by an operation. So she went back to a doc, and of course the rule said, well, you've had therapy that isn't standard practice in the UK, therefore we can't offer you an operation, even though the tumor had shrunk. And so, of course, front page article, and then things changed after that. Other personal experiences is Maurice, when his wife Josephine was diagnosed with cancer. He tried to access off-label treatments, a lot of resources, House of Lords, tried to access these treatments, really struggled to do so. And in the end, there's an act, we developed the Access to Medical Treatments Act. Why can we not do real-world therapeutic testing? What I'm talking about there is the drugs that haven't been tried in a clinical trial, but are there and potentially available. Why not test them in the real world, in a small group of patients, when patients have run out of options, rather than saying, no, there's nothing there. Join this. It is not a perfect clinical trial, so it's not going to cost a fortune, but we have this drug and we record the information in the health service. And it's a no-brainer, really. If you've got all these drugs and combinations, imagine you've got one drug to run a clinical trial across all different cancers, that's a thing in itself. What you have, if you have 2 drug combinations, well, that's already, we have 800 or 1,000 drugs to choose from. So 1,000 drugs already there. Have they been tested in all the cancer types? No. 1,000 times 1,000 is 1,000,000 if you take these combinations. What about 3 drugs? That's 1,000,000,000, but then you can vary the dose and the way you get the drugs. So you have infinite number of potential combinations. How are you going to test those? And how are you going to test those in an appropriate time frame? If it's not going to work in 10 or 15 patients, it's not going to work. Something's wrong. So why can we not move to something like this? So where are the barriers? Is it just the way we do things? And why can we not move to a health system where we learn from every single patient? We can pull information together in knowledge banks, run clinical trials when we have to, run an ecosystem where patients have access to new treatments based on some understanding and a hypothesis that it might work in that individual, and do it in a small cohort of individuals that can help us answer those questions. At least we can test them somehow, and a patient has an option when they previously hadn't had an option at all. So every patient without options now has options with shared costs. That's the place we want to get to. And how can we work together with industry to share this cost rather than having a big complete separation of the healthcare system and the payers for the healthcare system, the researchers, and then the pharmaceutical industry. And then when we learn on this, we're going to need large numbers, huge numbers if we're going to penetrate and understand the rarer types of cancers. And the more we understand about cancers, they're not, because of the diversity, it's really many, many cancers of small numbers each. So almost every cancer becomes a rare disease. So how do we address this? And the way we've started tackling this is through the International Cancer Genome Consortium. The International Cancer Genome Consortium was first formed in 2007, and that's when we had the technology to sequence the human genome, and we were ready to sequence cancer. So over the next 10 or 12 years till 2019, we sequenced 25,000 cancers, and we made them available with the TCGA internationally to be shared and used by researchers. And it's true to say that today no new drug or intervention is developed without using data from the International Cancer Genome Consortium. When the ICGC moved its headquarters to Glasgow, we redefined its next initiative, and that was really about patients, not about the structure and characteristics of the genome. We've learned most of that. I mean, there's a little bit to go, but we pretty much know it. But how does this relate in the important things? How can we use this to treat patients better? And our mission is 1 million patient years of precision oncology knowledge for the world. That exemplifies the time data that's important. It's not just a snapshot at this point in time. What's happened after a year? What's happened after 2 years? What's happened after 3 years? What's happened to that patient? Did they respond to this treatment? Did they not respond to that treatment? And we can reflect that back onto the genome. There's other aspects where you can start to follow the treatment of your cancer by what's called circulating tumor DNA, where DNA that's fallen out of your cancer is circulating in your bloodstream. You can measure it. And if it's going up, then you need to be a little more wary. If it's going down, you think, well, maybe my treatment's working. And you get an earlier readout. All of these things we need to study in more detail and change the way we do things. We're still stuck in the way we assess whether a drug works or not. We do a scan every 3 months and then go, oh, I'm not sure it's bigger or smaller. But we can do all these tests now. But the regulators are working on it. But at the moment, you can't get a drug approved unless you use standard criteria that you've been using for 40 or 50 years. There's a lot of people involved in the International Cancer Genome Consortium. This is slide's a little bit out of date, I just noticed. But we have 18 countries on board now. They're all contributing data. But one of the greatest challenges we have is data sharing. How do we share these data to address these sorts of questions that we need to know? This is Tessa. You may remember her from a few years back, who diagnosed with a brain tumor, suddenly discovered that there's all these things that can potentially be done that as a patient she hadn't heard of, off-piste, so to speak, and how these things are important to understand and then take the learnings from that arena and bring them into routine patient care. And I've noticed myself again, one of the first patients I ever treated with pancreatic cancer was a 40-year-old lady back in Sydney, Australia. You're young and you're really at the peak of your career. I did the most beautiful operation ever, if I do say so myself. She recovered beautifully, went home. Everything was fantastic. Looked under the microscope, had a tiny, tiny cancer, just maybe the size of my thumbnail, tiny, tiny. Everything was clear, the lymph glands were clear, everything was clear. And I went, I said, wow, she's going to do really well. 12 months later, she was dead. So do we actually know what we're doing? The answer is we don't. So we need to understand what we're doing, and not just on average, but for individual, these sorts of questions. And we all want to answer these questions, either as a patient or as a doctor or any other clinician that's treating people with cancer. What sort of treatment do I have? What sort of cancer do I have? What do I need treatment? We can overtreat cancers and we can undertreat cancers. In some circumstances, we go overboard and cause these side effects, but we don't know which ones we need to go overboard with and not. Others, like pancreatic cancer, we know the cancer is going to come back, yet we wait for it to come back till we can see it and go, oh dear, we need to do something, rather than saying, well, we know you're going to come back. We'll just keep going with another treatment if that's the appropriate thing to do. Can I access my treatment? We have all these rules, regulations in place about the average. We all pay taxes. We all contribute to insurance policy. We all do that. Rather than telling us, you can only have these treatments, A, B, C, and D, because we've decided, which probably in 2021 may well be against human rights. This is the contribution on average for a given patient for this disease. Within the realm of sensibility, with your doctors, you choose which treatment you wish to do. And there's precedent for that. So in the UK, postpartum, once you've had your baby, you can choose what postnatal care you get. Up to £3,000 worth is the limit, but still the precedent is there, the concept is the same. And of course, the specific, will the treatment work? What are the side effects? And of course, as I mentioned, how long have I got? So I'm just going to finish with this very important point. What is the point of privacy? What is the cost of privacy? We all want to keep our data. We don't want bad things like the Cambridge Analytics and other things to happen. Of course we don't. But what is it actually costing us as a society and as a community to keep our data private? I mean, to be honest, if as I get older, I'm going to blast all my diseases and my drugs all over the internet and maybe write them on my forehead. So if I go into an emergency department, people will know what I've got and they'll know how to treat me. I still remember as a youngster, a patient would come in and we go, what's wrong? What's wrong? Oh, we don't know. And you'd ask the wife, Mabel, what's wrong with Bill? Oh, I don't know. Did you take any medications? Oh, there's a little blue pill and a little white pill. And you go, oh, gee, I mean, what are we going to do here? And then we can't get the information. So then the patient dies. An hour later, the old notes come back, bang on the table. And you go, oh shit, maybe if I knew that, I may have done something different. Yeah. So what's the cost of privacy? Can we quantify it? No. But think of the advances we've made over the last period of time. Could we have not made them 20 years earlier, 30 years earlier, 40 years, 50? I would suggest that the cost of privacy is substantial, decades and decades. Of course, we can't just distribute data willy-nilly because there's a downside to that. But what is the right way of doing this, rather than the knee-jerk reaction that says, no, you can't have my data, and then at the end, what is the cost? So thank you for listening to me, and I'll be delighted to take any questions. Thank you very much.
Thank you very much, Andrew. That was great, and it was fascinating, and I think actually will really resonate with a lot of people in the SHINE community, because I think as younger people, so, you know, people who are in their 20s, 30s, and 40s, I think they have initially anyway, a lot of questions about why did this happen to me? So what is it that, you know, caused the mutations that gave me cancer in the first place, and whether those are the same as, you know, happen to older people. So we do have a couple of questions in the chat already. I guess I just wanted to kick off with a question of my own, which is really, so where are we now in terms of the tests that you talked about? So you were talking about, you know, patients accessing these molecule tests so that they understand exactly what type of mutations they have. How is that something people can get on the NHS? Do you have to pay for it? Is it widely available?
It's not available yet. It's people are working on it. Again, the limitation is what tests are currently required based on a drug approval. And so the moving from what we call single gene testing is currently happening, but it is just so slow. I mean, we have this concept of whole genome sequencing, which may work in some subgroups of patients like blood hematological malignancies or pediatric cancers, etc. But to do that, where you need big biopsies of fresh material, it's not part of the routine health service. In order for us to move from single gene testing to testing using these tests, there needs to be a philosophical change in that we're not just doing just what we're meant to do. We can do more for almost no extra cost, and that provides more information about our patients. And if you link those genetic tests with the molecular data, then you can get reams of information. And even if you can think about it from a business model sense, if you're acquiring those data as a health service, people who develop therapeutics will want to understand that so they can better develop their drugs. Of course, with all the requisite things in place, but that means that the patients are contributing to knowledge. They're contributing to the knowledge, which generates revenue and return for the NHS, and the NHS starts to sustain itself to a certain point. Does that make sense?
Yeah, definitely. I should have said, actually, just if you are on the chat, and I can see there's dozens of you there, this is a webinar, so we can't actually see you, but you can put any questions you have in the chat and we'll try and get to you. So I think just building on that question, Andrew, somebody's asked, okay, so we need this change. As a patient at the moment, is there a way that patients can volunteer to have their genomes or tissue sequenced or studies to contribute to research? What can we do as individuals?
Yeah, so it's very project specific now. Initially, when we first did ICGC, each country took on a particular cancer type. So we did pancreatic cancer and ovarian cancer, and it was whatever is important for a particular region. So the UK did breast cancer, and some others, some prostate cancer as well as a national consortium. At the moment, it's really part of clinical trials. So if you're part of that, and thank you for the volunteering and the volunteers that would contribute their information. There is the UK Biobank. I'm not sure what they're doing there at the moment. I don't have enough information about that. But there are certainly some initiatives that do require, love volunteers. The ones I'm involved with are specifically around clinical trials for new cancer treatments.
And we do have a session as well on Thursday afternoon, for those of you who are on this, with Leslie, who, Andrew, you mentioned, and a couple of clinical trials nurses as well. So if people are interested in finding out more about clinical trials, you can join us there. So somebody has asked, if I had a genomic test, what should I do now? What are the best steps to take if you have a rare cancer? And somebody else has asked a similar question about kind of targeted therapy for a cancer of an unknown primary. So where you do have a rare cancer, what would you recommend in terms of steps to try to find out as much as possible, make sure you are getting the best treatment?
Yeah, I mean, this is an important question. There's not a lot of people around the world that do this to understand that. So you need to identify whether there's a drug approved for your mutation or not, whether there's a clinical trial available that matches your mutation. Then the third step, whether the mutation is associated with a treatment response in a different cancer type, but you have the same mutation, but it's a different cancer type. And often companies will have, compassionate access schemes, etc. But it's a challenging path to navigate, and it's a combination of working with your oncologist to work out what those possibilities are. And that's the sort of order that you do them in. Is there something approved? If not, is there a clinical trial? Is there a clinical trial of this drug in some that's available? Maybe just not for my cancer, but often we do them in a group of cancers. Mm-hmm. And then, if there's a trial or a treatment targeting that particular mutation in a different cancer type, that gives you rationale to try and access that, and that gives you a reason. It stacks your odds against succeeding. It's not to say that it's going to succeed, but it's better than randomly pulling something off a shelf.
Yeah, sure. No, that's really helpful. So somebody else has asked, I don't know if you know the answer to this, but they haven't been able to get a biopsy from a bowel cancer that somebody in the discussion has. But is there another test that they could be pushing for so that they can find out what type of cancer they have, or more targeted treatment might be valuable?
Yeah, so these things are not available on the NHS, but circulating tumor DNA, the so-called liquid biopsy. So okay. The most accurate is, at least as we know at this point in time, is if you sample the cancer itself. But there are up to 20% of patients you can't get a biopsy. So either the thing's too small, it's such a sensitive spot. If you move by a millimeter, you're going to cause a lot of damage. But you can measure the mutations in the blood. It's not 100% sensitive because there might not be enough floating around. But if you find something positive, then it's helpful. But if it's negative, it may just be that it's below the level of detection. So it's circulating tumor DNA, or liquid biopsy, it's called.
Yeah. Carson, I can see you've raised your hand, but I'm not sure if I can unmute you. If you can hear me, do you want to type your question in the chat, and then we can get to it? Someone else has just asked. So, and actually we do have a number of people in the Shrine community who are kind of, I guess a bit like Leslie, who you talked about earlier, sort of, medical miracles. They people that you maybe wouldn't expect to be here, but because they've been on clinical trials, are, and one of them is in the chat. So Caroline, who said she's got a very rare cancer. She directly benefited from precision medicine and access to drug that's unlicensed in Europe via compassionate access scheme. So her question is, what as a patient can she contribute? How can she contribute to information sharing to make precision medicine more of a reality for others in her position? Obviously, she's one person and there's a lot going on, but how do you kind of cut through the noise and make sure that other people are able to find out about what's worked for you? Are there schemes we can contribute to?
Yeah, make a noise. Yeah, make lots of noise. Yeah, we need it's something that's like in the US, we call them precision medicine advocates, that would essentially compile these stories together and present them to government, organizations, health services, to say, well, the way we're streaming at the moment is never going to get to the point where you're going to help all of these people. So how are we going to change that? And particularly the nub of the debate is the privacy debate. And they're so welcome to hear to say, well, how can we help? Well, really tackle the privacy debate, which is, let's put appropriate safeguards in place, but let's not just quash everything because the risk of something happening is, for example, we can't share genomic data because there is a chance that somebody can look at the genome, measure your genome, and then identify who you are on a database. I wish somebody was so interested in me that they would do that. What a load of effort that is. It's just an unrealistic fear, but we're sitting at this unrealistic fear component. There's some legitimate fears and legitimate concerns, but we're sitting so far up this end, and the only group of people that can shift that will be patients. And they have to be heard because all these committees, all these other sort of public organizations, many of them are not, I shouldn't say this, but many of them seem to have lost their path. The concern is always on the path of policy, de-risking, no risk, zero risk. If we take zero risk, there's zero gain. So what is appropriate, an appropriate risk as a society and as a community that allows us to really glean those data together? Various initiatives like having all your medical data on your mobile phone, sharing it with databases with appropriate safeguards, but really pushing that from a patient perspective, saying, we want this, we want this, we want this information to go out and to really benefit and advance what we're doing for cancer.
Yeah, absolutely. I think that's a really important point, because one of the things we're very passionate about at Shine is patient involvement. So we're a patient-led charity. Everyone that works for us has had cancer. Almost all of our trustees have had cancer as well. But that's not just a kind of a nice to have. I think it's really important. I think some organizations, the British Medical Journal, for example, has done a lot of interesting work around trying to involve patients in research as well. So for everyone who's on this call, I think keeping an eye on how you can be involved in all of those discussions. And certainly data, I think, is one of those that it's not well understood. And then people just freak out and think, oh, I don't want to share my data. But actually, like you said, a lot of patients are really would welcome the opportunity to do that, because they want to help themselves, but they want to help other people as well. And I think that's very much underestimated by the medical community. A couple of other questions. So somebody has said, I'm just going to read this. Well, someone's also said thank you to you, Andrew, because you've helped her to understand why she was receiving the treatment being offered, and your slides have explained it to her much more clearly than her team did. So that's great. But she's also asking, how soon do we think this kind of medicine will be, or this kind of testing might be available on the NHS? I mean, are we talking decades, years, months?
It depends on how hard you guys push, is the answer. Patients demanding, we want this, will change thinking, because at the moment it's top down. Oh, do we have the evidence to do this? No, we don't have the evidence to do this. No, we don't have the evidence to do that. If patients were saying, somebody that was sharing her story said, well, I found the right drug for that, they're the things that need to come out front and center and say, well, look, if I'd gone down what you were giving me on the supermarket shelf, I'd be dead. But look what happened. And we understand we can't do this for every single patient, but the very concept of that you're now testing for all these genes and you're doing different individual tests. Each one of them has a different protocol. You need to employ a different person for each one of these. Why not just put them all together and do one test? It's the same test. It's standardized across the country. And then drug developers, drug companies will invest to say, well, hold on a minute, if you're doing this test, we'll help you pay for that test, because it saves us paying full whack for the test if we run the clinical trial. So let's make a deal here. Let's say, well, we've got the drugs, we need to develop the drugs. Your patients want new treatments. Patients want new treatments. Doctors want to give them new treatments. Drug companies want to develop their drugs, want them to get them to patients. But there's some sort of impasse. I mean, what's the bit in between that's blocking it? That's why I asked the question, I mean, are we the problem? Are we the problem in the interview? I mean, on one side, you've got all patients that want new drugs, doctors that want new drugs for their patients. You've got drug companies that want to run clinical trials and they can't get patients onto the trial fast enough. And yet there seems to be this impasse in between. Is it the way we've started conducting clinical trials? It could take 2 years to set up a clinical trial in cancer. Just think of what happened in COVID. 6 weeks. So if we can do that in 6 weeks, why the hell are we going to go back to spending 2 years to set up a clinical trial for cancer? It just doesn't make any sense. If we can do it in 6 weeks, and the number of more people die of cancer every year in the UK than they've died of COVID over the whole period of time. I think there's been 150,000 deaths of COVID over 2 years. 165,000 patients with cancer die every year. I'm not saying COVID isn't substantial. It's important, but the relative thinking is that, oh, COVID, oh, geez, viral endemic, bang. We have a cancer pandemic every year, every single year. And we don't think about it as a pandemic. We think, oh, yeah, well, we expect 165,000 people to die every year. That's all right. Whereas, oh, gee, 80,000 people died of this virus. Oh, dear. And the average age of cancer deaths is lower than the average age of COVID deaths. So we need to readjust how we think about these things as a society, and not be so reactive.
And so that actually leads on to a question that someone else has asked is, given all that change and progress we've seen through COVID, right? Setting up clinical trials in 6 weeks, getting a vaccine in less than a year. Do you think that things will change? Do you think it will kind of open people's eyes to making faster progress in cancer drugs, enabling people to roll out clinical trials more quickly?
So, I think from the clinical trial perspective, I think, yes, there will be some progress. But the major, if you think, the biology and the science is not the limiting factor. The limiting factor is all this other stuff that we've created. So one of the things I do is, with a team of 20 of us in a company called Keynote Oncology, we look for different drugs around the world that aren't being developed for whatever reason. They've been shelved. And we look at them and we say, can we actually take this and can we develop it so it's going to be useful? And the science usually stacks up really nicely, but the patent is somehow wrong. So you can't take it on because nobody will invest in your company to do so. Somebody's made an assumption in something or other, or made a deal in the previous terms that it's just impossible to take this drug forward. So most of these impediments are not in the science. Most of the impediments are things that have just happened over time through misalignment of thinking and misalignment of systems, or because they've been forced to do it. It was essentially, well, I'm never going to get this drug to see the light of day unless I make this deal here. And then that deal becomes unpalatable further down the line. So that's the point that I made about in the little video, that most drugs never get developed because they just get stuck. And they get stuck because, well, if there's no pathway forward in a business or a commercial or a societal sense, there's just no pathway, even though the drug's probably pretty good. It might even be better than the ones we have now, but we can't, it's stuck. Does that make sense?
Yeah. So we need to unstuck it. We need stick it, not unstuck it. We need a revolution then in the bureaucracy surrounding all of this stuff.
Yeah, bureaucracy, it's patent life. It's more complex than just bureaucracy because it's also how drugs are approved on what basis. You think about the HIV movement. What made all of that happen was patients. That's what made all of that happen. And there are more cancer patients than there were patients with HIV back in that day. But we're set in that thinking that, well, we sort of accept that 165,000 people die of cancer a year, and we should change that thinking. We just, make some noise. I can make a lot of noise, but they don't listen. So.
Well, that's all right. I think you're probably going to have a lot of people contacting you going, right, I'm ready afterwards. So we've got a couple more questions. So somebody's asked specifically, and this is their question. I've had genome sequencing for an osteosarcoma, but was told they couldn't do full sequencing. Is there any reason why?
I'd have to understand the detail of what they were looking, what they used for full sequencing. He may be referring to whole genome sequencing. So this is so whole genome sequencing is great for research. It's not quite ready for the clinic yet because you need a big piece of fresh tissue. And if you don't have enough cancer cells within there, you're not going to detect the mutations because every tumor is composed of cancer cells and normal cells, what we call the tumor microenvironment. And if you don't have enough cancer cells, there's mostly cells that are normal within that mass, you can't detect it as below the threshold of detection. So that's probably the explanation, not knowing the detail.
Another question is about if you are looking for clinical trials, do you have any thoughts on where people could go specifically, to find out more about clinical trials? Obviously, some hospitals have clinical trials nurses, so I guess that would be one step they could take. Are there other places that they could go to look more widely outside of the UK, for example? Yeah,
so I mean, this is a big challenge. In fact, I had a company I started years ago on this called Cure Forward. You can, of course, go yourself with clinical trials and do an internet search. You can get your doctor to do that. that can be challenging unless something pops up immediately. There are a number of companies that work on this. There's a company called My Tomorrows, which is a global company, but it's based out of the Netherlands. There's a company called Citizen, Citizen, on the west coast of the United States. There are a few more companies that I don't know what's happened. There were patients like me, etc. I think they were acquired by other corporations. But it's, yeah, there's My Tomorrows. I did actually just do some work for in the past, and they've grown, and they've got a good model. So I would suggest they contact them.
Yeah, and actually they're really interesting because they will, I think you put in all of your details and they can produce a report for you, and some of it might not be relevant, but some of it could be, and it's a global search, I think, isn't it?
It is, and I know the founder, a guy called Ronald Bruce, and he started that because his father had lung cancer and he couldn't access the drug that he wanted to, or he couldn't navigate the path, and so that's why he founded the company.
Yeah. So one other question I had, which I know it comes up for a lot of people in our community. Obviously, cancer occurs because there are mutations, and other people who have spoken to Shine have kind of said, well, typically it happens in older people because that's when your cells start to change and you get these mutations. So if you're looking at people in their 20s, 30s, and 40s who have developed cancer, I mean, we've heard kind of varying things, I think, where people say, well, actually, you're too young for maybe lifestyle factors to cause these mutations. But do you have any sense of why younger people would get cancer when, they're not old, their cells aren't breaking down, they're not in their 80s? What is it that causes cancer in younger people?
Yeah, so if you think about, we're talking about younger people now, not neonates and children, we're talking about younger people. So this is probably not a good enough answer, but I'll try and develop it. It's called the motherhood hypothesis, where the right constellation of events happen. And so your risk of getting some of those events increases as you get older, but it doesn't mean your probability of those events younger don't exist. And there are some inherited cancer genes that can cause that. There are some probably rarer inherited cancer genes that we'll never understand because they're too uncommon to know whether they actually predispose. But it's probably just the likelihood that, you can have mutations all through your genome and it hasn't hit the right genes to cause a cancer, or it's hit them in the wrong order to cause a cancer. And we know that there are some things that look like a cancer, like molecularly, but don't exactly behave like a cancer. But there's always the chance that you would just have the wrong mutation in the wrong gene in the right order, which will trigger something. That's probably an answer, but there's no proof to support that. So, but I would agree that the environmental exposure increases your probability, but it doesn't mean that you can have one cigarette and you can have a mutation in the right gene that causes the cancer versus smoking for all of your life and it's just happened to miss your gene.
Yeah. Thank you. Okay, a couple more questions. We're almost out of time. So someone said, what are your options when you're told you can't access clinical trials because your cancer doesn't behave in the normal way? Does cancer behave in a normal way?
Tell me what normal is. Yeah, well, it's not in the normal way. It's because it's the way they've set up the clinical trial. We often, when we're negative about it, say, well, you can't get into this clinical trial unless you've got one green eye and one blue eye. And it's the only way you can get in. And this is what I was talking about, the regulatory components of how you do this and how we can move to something like expanded access programs or therapies outside clinical trials, so that you can when you're developing a drug, you don't want to take any risks in getting the wrong patient groups in that drug when you're developing it, because then it'll fail. But what you can do is have an expanded access program sitting in parallel to that. Say, well, this is the trial, this is where we're going for our approval, but we want to see whether this drug will also work in patients that don't quite fit into these inclusion criteria and exclusion criteria. But then it's not, if we go to file for regulatory approval, we're filing this and we don't have to file that because it's a different group of patients. So it doesn't need to be run as a formal clinical trial. And so there's examples of that where they've done that, and then they've done the less matching cohort of patients that the ones that don't behave like they're meant to behave, so to speak. And that addresses the question there. So that's one of the challenges, that their inclusion-exclusion criteria are too rigid. But there's good reasons to do that from a drug developer's perspective. And it's driven by the regulators to a certain degree. They're trying, how do we do this? And then what is the cost-benefit analysis for the payers after that? And how does all of that work, and how does that fit in together? It's a bit too siloed at the moment. It needs to be more streamlined.
And so if you were in that situation, would it be worth looking at, I don't know, so say I had non-Hodgkin lymphoma, you were interested in that, you could look at what clinical trials are available and see if they did have an expanded access arm to those trials that you might be able to join.
In fact, now on clinicaltrials.gov, just starting a couple of years back, there's an expanded access program record as well. So it's not just a trial, there's expanded access as well.
Great. So final question, probably. Somebody has asked, and this is an interesting one, is it possible to have genomic sequencing a few years after going through treatment? So at the moment, if you have a biopsy, does that get saved? Can you go back and look at, what went wrong so that you've got that knowledge bank for the future?
Yes. It depends on each health service, how long they keep it for. Okay. But if you get a biopsy, generally it's still in the pathology department unless it's been exhausted. And you can go back to look what it was then, but we also know that cancer treatment can change the characteristics of a cancer. Not all the time, but some of the time. It's just called a selection pressure. So if you're attacking a cancer in one particular thing, it's like an army will respond by changing its position or changing its composition. And it's called like induced resistance. So we know if people have had, say, a therapy called olaparib, the BRCA1 can revert itself to basically save the cancer. And it's astonishing how smart these cancers are, so to speak.
Okay, but that's good to know that we can go back as well as maybe sequence what's going on now. Andrew, thank you so much. This has been really fascinating, and you've, I think, given everyone a huge amount to think about, but also hopefully kind of fired us up to fight as patients for expanded access and change to how information is shared and how we can access those drugs. So we really appreciate all of your time. And if anyone does have any other questions, you can feel free to message us through the Shine Cancer Support, the Shine Connect portal, and we can try and get those answers for you. But for now, that's it. And we will hopefully see some of you at other sessions. So thanks so much, Andrew. Have a good day.
Thank you very much, Karen. Thanks, everybody.